Hey guys, today we're going to talk about glycolysis and more importantly about the details. So in our last video lesson, we had an overview of aerobic cellular respiration. And as we mentioned, there's 4 stages. Gotta get used to this. 4 stages, and we're going to talk about each one separately. Today in class, was where we looked at the chemical structures and the enzymes involved, and you put them together like a puzzle. You figured it out based on the starting material, knowing it's glucose, and the ending material, knowing that you were getting 2-pyruvate. So let's look at the details together. So first of all, glycolysis is thought to be the most fundamental and most ancient of all metabolic processes. Understanding the fact that cells need energy to survive for any reactions to occur, so they need a way to be extracting energy from their food. The supporting evidence that suggests this is glycolysis is universal. And when it is said that something is universal, it means it is occurring in all or most organisms. The second piece of evidence is that glycolysis does not require oxygen. And this is important for 2 reasons. One is oxygen wasn't really present when life began, so it became abundant 1.5 billion years after life began. So it wasn't even available for this process to occur. The other part is not all organisms we know can survive with oxygen. So think about what those are called again. And thirdly, glycolysis occurs in the cytosol. And we know that not all organisms have membrane-bound organelles. So all organisms have cytosol, but if glycolysis occurred in the mitochondria, maybe glycolysis evolved after organisms evolved membrane-bound organelles. So those are 3 reasons. You may be asked those as a multiple choice, or you might be asked to explain why glycolysis is thought to be the fundamental metabolic pathway. So here we have 2 graphs to look at the different steps of glycolysis. And I put 2 graphs not to confuse you, but to actually help us a bit more with the understanding. Together they make a perfect graph. So you will actually often, and I want the left graph is actually a little deceiving and harder to follow, but the left graph is actually more common what you will find. So the one on the right is actually really hard to find. You won't see it too often, but it actually explains it a little bit better. So keeping the 2 together, we'll start looking at them. So I'm going to refer to the one on the right, which would look like this to you guys. The one on the right. We have glucose going in, and again, 6-carbon molecule. And immediately we get a phosphorylation reaction occurring, so a phosphate group coming off of ATP. So we get the production of ADP. So energy in. And we get glucose 6-phosphate. So this is telling us that the phosphate goes onto the 6-carbon. And then more on this in first year university. We get an isomerization reaction from glucose 6 to fructose 6-phosphate. So same chemical formula, different structure. And then we get another energy in where we get fructose 1,6-biphosphate. And with this, we have, you could guess, the phosphate group is on the 1st and the 6th carbon. So we have energy in. So here's our 2 energy in reactions. From here, fructose 1,6-biphosphate is fructose 1,6-biphosphate is split into 2 molecules. So I want to shift over to the left graph here because the right graph doesn't show it so well. So we get G3P being produced, which will continue down in the reactions. We also get DHAP produced, which then gets converted to G3P. Okay, so it's just indirectly being made. Okay, so in this graph on the right, we see DHAP getting converted to also called GAP or GAP, but we call it G3P. Okay, and then again, another molecule being directly converted from glucose, fructose 1,6-biphosphate, G3P. Okay, so ultimately you're ending up with 2G3P. This graph is nice because it's showing both going down. Most diagrams you will run into go like this one, and it's just telling you 2, 2, 2, 2. But it's nice to see where this 2 is coming from. So we are going to continue to talk about the graph on the right. So each G3P is going to get oxidized, losing hydrogens, losing electrons, and that reduces to NADH plus to NADH. Those two NADH are going to go to the electron transport chain, and we're going to get conversion of NADH to some ATP. Okay, so this is energy being produced. Then from there we have the 1,3-bisphosphoglycerate, 1,3-bisphosphoglycerate. And this is nice because this is a short form to see that. And now we get production of ATP. So we have substrate level phosphorylation occurring twice. So we have 2 ATP being produced here because we have 2 of these molecules. And then we have 3-phosphoglycerate here. We have an interesting reaction called a mutase reaction, where you just have the phosphate group moving from the 3rd to the 2nd carbon. And then we have a loss of water. And there is electrons being moved from one part of the molecule to another, so it's called a redox reaction. And we get the production of 2-phosphoenolpyruvate, PEP. And then the phosphoenolpyruvate leads to pyruvate. And again, that creates 2 ATP molecules. So those are the steps. If we count these, we have 1, 2, 3, 4, 5 steps where only energy has gone in. Those are the first 5 steps. Then we have 1, 2, 3, 4, 5 steps where we have the production of energy either directly through substrate level phosphorylation. I was getting them mixed up. And I wanted to say them together. And then we have the production of NADH, which will produce ATP through oxidative phosphorylation. There are 3 key features that your textbook points out, and so we will go over those together. So the first 2 ATP are consumed as glucose and fructose 6-phosphate become phosphorylated. The 2 ATP are used to increase free energy to allow the other reactions to occur. Even more free energy is released in the payoff phase because you're getting 4 ATP and 2 NADH molecules. So you have 2 ATP going in, 4 coming out, plus the 2 NADH. The 2 net, so key feature number 2, the 2 net ATP, and so net ATP meaning 2 went in, 4 came out, so the difference is 2 being made. So you're getting 2 net ATP and 2 NADH for every glucose that goes into glycolysis. It's important to remember no carbon is lost. So we are just getting a 6-carbon glucose, and you're getting 2 3-carbon pyruvates. We also see that water molecules are produced. A lot of times you won't actually see this in the reaction because they're actually used in the hydrolysis of the ATP in the beginning of the reaction. So it kind of isn't really necessary to include those in the net reaction. They get produced, but then they get used up. The third key feature is the production of ATP through substrate level phosphorylation. So we have enzymes that are transferring phosphate from a high energy molecule to the ADP that is available from the cytosol. This is the mode of ATP synthesis that is used during the citric acid cycle as well. We also see NADH being made, what we talked about previously, and we will see how that leads to the production of ATP. So the net equation, and again, net is only talking about the important things that go in and the important things that come out. So we have 1 glucose going in, and we have 2 NADPs going in and 2 inorganic phosphates going in, and 2 NADH plus going in. The glucose essentially is broken into 2 pyruvates, and then we have our 2 ATP coming together, being produced, net, because we know 2 go in, 4 come out, but a net 2 ATP are being produced. And we have 2 high energy electron carriers, 2 NADH being produced, and then 2 hydrogen ions being produced, which we will see the importance of those in a future lesson. So I hope that summarizes the basic overall steps of glycolysis for you. And then next, for our, I believe, last slide or second last slide, we are going to talk about the energy produced from glycolysis. So energy stored by 2 ATP is 62 kilojoules. So the net reaction of glycolysis produced 2 ATP. So we have 62 kilojoules. However, if we completely oxidize 1 glucose, we would be getting 2,870 kilojoules. So you're looking at 2.2% energy, available energy from the glucose being produced in glycolysis. Not very effective. And we'll look at how ineffective that is in the end, and how some organisms really just rely on glycolysis. So not a lot of energy coming out. So let's talk about why we still do this, how we survive. So for every molecule of glucose, you're only getting 2.2% of the available free energy being converted to ATP. So again, not much. If you're getting 2.2% out of something, you're not doing a great job. So some energy is being lost, thermal energy, sure, that happens. But most is still conserved in those 2 pyruvate molecules and those 2 NADH. Again, we remember that those 2 NADH are going to be leading to the production of ATP. But those 2 pyruvate have those 3 carbons, so there's a lot of energy in those bonds. So those pyruvate molecules are continuing to the next stage, where we get the pyruvate oxidation, and then more importantly, we get the citric acid cycle, where we're going to draw more energy out of there. So more to be seen, more energy to be extracted from those 2 pyruvate molecules. Tomorrow we're going to look at some of the next steps, so the pyruvate oxidation. It's a quick one, we'll look at that and then we move on.